какой-то век я в время попала. Я опоздала. Спасибо большое. Good morning, guys. We start in time. So not as usually, the weather is very fine. So I get in time to my work. So today's lecture is about viruses. We're supposed to discuss viruses of humans and touch a bit bacteriophages, the viruses that infect bacteria. So I hope you hear me very well. If you don't hear very well, let me know. Guys, start coming. I hope you have something warm to drink in front of you. And as I do, and we can start now. And guys, start coming up. Guys, start collecting. Good morning, guys. Where are you? I don't see anyone here. My dears, where are you? Start coming. We have started already. Okay, I won't wait because I don't have time. Then later on you take a look at this lecture. So at the very first slide of our lecture, you can see a picture of the disease caused by the first discovered virus, that was tobacco mosaic disease virus. That what it causes, you can see here, to the leaves of infected plant, to infected plant organism. And here you can see the appearance under the electron microscope and the very sophisticated structure and the way of assembly of the tobacco mosaic disease virus. So, but of course, I would like to remind you that while we are studying not just virology, but medical virology as a part of medical microbiology, we will be talking today not about plant or whatever viruses. We will be talking about medically important viruses features. So, and then we'll continue this in special virology next on. So today we just take a general look at the classification of the viruses, their structure, how they multiply or how they replicate, and what tricks do we need to grow them in the laboratory to perform so-called culture. Guys, I don't see anyone here. So then I would like to send a message about the lecture. Maybe you've forgotten about the lecture. Nobody is coming. Guys, let me know who are coming. Okay. Oh, Chandor, very good that you are here already. So start coming. Good morning or good afternoon, Louis also. Okay. So I have to mention at least one name, and this is a name of Dmitry Ivanovsky, the researcher who in 1892, working in the Nikitsky Botanical Garden in Crimea, right here. So I hope you visited Nikitsky Botanical Garden. It's on the halfway from Alushta towards Yalta. So he had been working on the tobacco leaves disease. So, and then he did a lot of experiments to know what kind of microbe caused the disease in tobacco in tobacco plants. So, and he measured the infected plants and add physiological solution and filtered this mesh through the antibacterial filter. So he's supposed to understand whether the infectious agent is very small or cannot be stopped by antibacterial filter, but it is appeared that it cannot be stopped by antibacterial filter, cannot be trapped. So that's why initially Dmitry Ivanovsky called the tobacco mosaic pathogenic agent as filtrable agent due to the filtrability of the agent through the antibacterial filter. He did a lot of other experiments with the supernatant or with the filtrate that's supposed to contain this infectious agent. And he revealed that it will be destroyed by boiling, but it will not be destroyed by very long incubation in this filtrate or freezing, or then he showed that the filtrate from this filter is infectious because he poured this watered other plants with this filtrate, and they get sick. So, but then he didn't give the special name for this infectious agent. He generally called it filtrable agent. Initially, he thought maybe that was very small bacteria, and he tried to culture it on the bacteriological media, but nothing grew there on those bacteriological media. Then he thought maybe this is something new they never had and heard before. In 1899, the Dutch microbiologist Martinus Beijering, who also had been working with the tobacco mosaic disease agent, he found the way how to reproduce these infectious agents, and he understood finally that they are intracellular parasites, those organisms, they need the living cell to live. And then Beijering used the term virus to refer to the very specific pathogenic molecule incorporated into the cell. So these two share the priority of discovering viruses. Now we look through the general characteristics of the virus. So they are the infectious agents that do not possess a cellular organization. They are not cells, and they cannot be seen under the light microscope mainly. So, and last time we have discussed the way of living things classification. But that classification, I would like to remind you, was the one for cellular organisms, and finally viruses had been excluded from this classification because they are not cells. They have absolutely different structure. As you see here, they don't have any organelles and cytoplasm that is typical for cells, because eukaryotic cells and prokaryotic cells, they are not identical, but the plan of organization is same. So then the very important feature of the viruses that they may keep their genetic information in DNA or RNA. So you have to remember and know that all other living things keep and cellular organisms keep the genetic information in DNA. And RNA is used just for some technical assistance of DNA expression, like messenger RNA, transport RNA, but cellular organisms don't use it to store genetic information. But then in viruses, they may keep their genomes made up of DNA or RNA. So, but then these two molecules are usually not found in the mature viruses. But then they usually have over the capsid. That is usually called envelope or supercapsid. It is made up of bilayer of lipids, and usually this membrane lipid bilayer is taken from the host. So this is not virus specific. Then I would like to underline that if the viruses are completely built up, mature, and released from the host cell, so and ready to infect a new cell, so in that state, they may be referred to as virions or virions. So how to answer what is a virus? This is extracellular infectious virus particle. Of course, we may call it virus also, but to underline that this is an extracellular stage of the virus life cycle, we consider it as a virion or virion. Then viruses have some general reproductive distinguishing features. So, they are obligate intracellular parasites, so they may multiply inside the living cell only. Why? Because they cannot generate energy by themselves, and they don't have biosynthetic machinery like ribosomes. They don't have any organelles to reproduce their own protein. So that's why they have to take over the biosynthetic machinery of the host cell. So, and then they have the unique method of replication that is called replication. Why? Because they cannot divide the virion when it enters the host cell. It cannot divide. It is not divided. So, while the other living things, the cells, they multiply due to mitosis or meiosis. So viruses have absolutely different way. When one virion enters the host cell, it managed to make hundreds or thousands of identical replica of initial viral organism. So that's why of replica, this identical copy. This unique method of multiplication is called replication, when out of one organism, thousands of progeny viruses, offsprings are produced. So then, one more very important feature of the viral replication is that the components of the virus or virion are produced in different cell compartments. For example, in some viruses, so genomes of the viruses are transcribed and copied in the nucleus. In some viruses, usually RNA viruses, this can be done in the cytoplasm. Then the proteins are produced usually on the ribosomes in the cytoplasm or the cytoplasm. So that's another compartment. And some proteins usually undergo post-translational modification on the endoplasmic reticulum, EPR, right? So there's another compartment where we have the upgrade of the proteins, so glycosylation of the proteins and so on. So, but then, from different compartments, then the parts of the further virion will be transported and assembled out of individual components to produce an infectious virus particle. So this way is called disjunctive way of replication. And straight after the assembly, the infected cell usually release these virions, and then sooner or later the infected cell where they replicate gonna die. So then if it dies very quickly, straight within the virus release, so this virus is called cytolytic because its replication causes the death of the cell directly. But some viruses are milder. So they are released by the cell and the cells stay alive for a while, right? So it doesn't die immediately. So, but then within several days, it's gonna die anyway. But those viruses that don't cause the immediate death of the cell are called non-cytolytic. Of course, they cause harm by their replication, they cause harm to the cell, but not that fast. Okay, how can we give names for the viruses and how can we classify them? Several features of the viruses are matter in classification. They can be classified or categorized according to the size and morphology. Then they can be classified according to the structure. They can be more complex and less complex. Then this is very important that they classified according to the nucleic acid they use as the genome and the mode of replication. We'll take a look today at those classifications. But also, what can matter in naming of the viruses, for example. This is a sort of disease they cause because we have a large family of so-called hepatitis viruses, around 6 to 8 different types of hepatitis viruses. And we also may have viruses that have that are neurotropic and cause encephalitis. For example, West Nile encephalitis virus, right? So the disease is also important in naming. Then sometimes the way of or the route of transmission is important. We have a group of viruses that can be spread through arthropods, like vectors. These are the blood-sucking insects that are called arthropods. So this is a group of so-called arboviruses, arthropod-borne viruses, right? So then sometimes in the names of the viruses, we mention the tissue tropism. For example, we have a very large group of so-called enteroviruses that may replicate in the enteric mucosal layer, epithelial cells. And by the way, poliovirus, that the cause of poliomyelitis, is an example of this enterovirus. Then the host ranges matter. So we may categorize viruses according to their hosts. But as far as we see now, there's not so big difference between human and animal viruses. Some animal viruses don't cross intracellular barrier at least by now, But coronavirus and HIV virus, they give us very good examples of how the viruses may cross the interspecies barrier. So we have plenty of those viruses, dengue, Japanese encephalitis viruses. So many of them can be transmitted from animals to humans. And generally, they are animal viruses. So that's why we consider them in one group of animal and human viruses. But apart from animals, viruses may infect plant cells, but also that is important for medical microbiology. Some viruses may infect bacteria, and those viruses may lead to the bacterial death. So those viruses are called bacteriophages. And sometimes they can be quite useful in medical microbiology. For example, we may use them to treat bacterial diseases because as soon as these bacteriophages infect bacteria, they kill them, but they are harmless to humans. So, first we take a look at the morphology of the virus's size and shape. So you can see here that host cell is often 1,000 times the volume of a virus that is infected it. So you can see, for example, the erythrocytes as an example, and these are the viruses, and this is the E. coli cell, right? So that is generally compatible with the human cells in size. So the size of E. coli is average 1 to 5 micrometers. So the human cell 30 to 50 micrometers. So we measure it in micrometers. So the size of the virus range from 20 to 30 nanometers or 0.02 to 0.3 micrometers in diameter. The largest one that makes 300 nanometers in diameter can be seen under the light microscope if you stain it by as a dot. That's it. So, and we know that mainly the only vaccinia or smallpox virus can be seen because that is the largest one. The others won't be seen. So, and the rule is that the larger the virus is, the larger is the genome. So, these viruses are generally more complex because they may produce more proteins from their genome. And according to the shape, they are usually quite often they are spherical. But we may also see the rod-shaped tobacco mosaic viruses. We may see the bullet-shaped rabies virus. So, but then you should understand that the shape of the virus, that what we see under the electron microscopy of the virion, the extracellular virus particle. Because as soon as the virus enters the host cell, it disappears as an infectious agent. The only nucleic acid and some proteins will be participating in the replicative cycle. So then, according to other shapes, so the pox viruses are brick-shaped, and we may also have a group of filamentous viruses that are called filoviruses, thread-like structures that, and the example is very well known, especially for Nigerian and other African students, that is Ebola virus. So, but then, of course, this is more important that the classification of the viruses can be based on the structure. And the simplest structured viruses are called naked viruses. Their nucleic acid that is black inside is covered by the protein coat that is called capsid. You can see here. And the capsid with the enclosed nucleic acid can be called nucleocapsid. The capsid is composed of usually large number of the repeating identical morphological subunits called capsomers. So each capsomer is composed of one or several proteins. It's very important because it helps virus to save the volume in their nucleic acid and their genome for some other proteins, because the space inside the virion is very small, so they need to save their genetic information. So the same proteins may be repeated many times in making the capsid. Viruses that are covered with one layer, that is a capsid, are called naked or capsid viruses. But some viruses of this type can be still very sophisticated. So some viruses, especially RNA viruses, need to protect their RNA inside very well. So they may build capsid made up of several layers of the proteins. For example, rotavirus has 3-layered capsid, but still it is called naked virus, because that is just the very complex capsid that covers the nucleic acid. So the capsid that protects the nucleic acid can be of different symmetry. So, and we divide them according to the type of the capsomer's arrangement. So, for example, these are two typical forms. The first one is icosahedral symmetry, and the second type is helical symmetry. This is the capsid and this is the capsid also. But then bacteriophages give us an example of the complex symmetry, because the head of the bacteriophage has the capsid symmetry, and the tail of the bacteriophage, through which the nucleic acid will be injected into the infected cell, has the helical symmetry. So that's why we call it complex type of symmetry. So this is an example of typical naked icosahedral virus, so with icosahedral type of capsid symmetry. And this type of symmetry is typical for naked viruses. So in a group of human or animal viruses, then there are no viruses that are naked and have helical type of capsid symmetry. They just don't exist, because it means that in the helical type of symmetry, the protection of the nucleic acid is not enough for the virus to survive. So, and the icosahedral or cubical symmetry have the capsomers, which are usually arranged in triangles, you can see there. And these triangles form a symmetric figure that is called icosahedron, with the approximate outline under the electron microscope of a sphere. Adenovirus is a very typical icosahedral virus. Okay, but not all viruses are that simple. So another type of the virus categorized according to the structure is the type of enveloped virus. And the example is influenza virus. Except for the capsid, they have an extra outmost covering that is called envelope. And the envelope is made up of lipoprotein membrane that is taken from the cell membrane, and it composed of cellular lipids, and it is derived from the host cell. But then this lipoprotein membrane is modified with virus-specific portions. So, protein, sorry. So we have two portions in the envelope. the lipoprotein membrane that is cell-specific and the virus-specific protein or proteins that are virus-specific. Those proteins that are incorporated into the envelope usually are glycosylated proteins or glycoproteins, and they usually form spike-like projections on the surface of the virion. So, some enveloped viruses may have one type of the glycoprotein. For example, like some Hepatitis viruses, or they most probably may have two or more types of glycoproteins. For example, herpes viruses, they have from 11 to 13 different types of surface glycoproteins. So you can see here the structure of the enveloped virus, and the influenza is given as an example. You can see the nucleic acid inside covered with the nucleoprotein that is bound to the nucleic acid to make a nucleocapsid. And outside there is the supercapsid that is called envelope. And the envelope is made up of phospholipid bilayer that is white here, and two types of glycoproteins that are polymerized to form spikes. So, and here you can see two types of spikes. They are colored in different colors, right? So you may have hemagglutination spikes that are made up of three protein hemagglutination subunits to form a spike, and neuraminidase spike that is made up of four neuraminidase proteins polymerized in a particular way. But then this type of enveloped viruses usually have an extra layer that lays in between the envelope, here white, and the nucleocapsid. And this layer is made up of M protein or matrix protein. And the importance is the matrix, that is matrix protein, mediate the morphogenesis or assembly and maturation of the virus inside the human cell. So we'll be talking about human viruses. So sometimes we may have some enzymes bound with the nucleic acid inside. That's more typical for RNA than to DNA viruses. So then the question is, how do you think whether the naked viruses that we have here or enveloped viruses are more sensitive to the different environmental conditions, drying, for example, or some disinfectants and organic solvents? It appears that the naked viruses are more stable and more resistant to all these factors. Between they have very tightly packed protein shell, and it is not easily disarranged by the lipophilic agents or dry. While the enveloped viruses appear to be very sensitive to the detergents and the drying and desiccation and lipophilic solvents, because this envelope layer is very fragile, like the membrane of the human cell. So then it can be easily dissolved or disturbed. So, and if the virion will lose these spikes on the surface of the envelope, it will not be able anymore to enter the host cell. That is the thing. So that's why those viruses are not more sensitive like all students think, because they have two layers instead of one. No. They are more fragile and more sensitive to the detergents, desiccation, and organic solvents. But all viruses are sensitive to heat. If you boil them, they all will be inactivated due to the protein denature. So you can see that in influenza virus like this and parainfluenza virus, the internal capsid has not icosahedral bihelical structure or helical type of symmetry. So, and there is no box-like structure inside. But the molecules of the proteins, of the capsid protein that's supposed to protect the nucleic acid, they bind tightly with the nucleic acid. Could you see here? This is a photograph. So the black one is the RNA genome, and the white subunits are capsomers bound tightly with the genome. And because the nucleic acid is typically helical, so the capsomers of the capsid follow the structure of the nucleic acid. So the symmetry of this type of capsid will be helical. So, and we have many examples of those helical structures, like measles virus and others. So, the function of the, now we discuss the function of the different proteins of the virions. If we take a look at the naked virus, the capsid proteins have protective function. It means they protect the viral genome from being inactivated. And the second function is the receptor function, because they attach, and that is the initial stage of the viral infection. They attach to the cell, human cell surface receptor. This is a membrane receptor that virion uses to enter. So they bind with this receptor by the capsid proteins. So the second function of the capsid proteins in the naked viruses is the receptor function. They bind with the receptor that allows the virion to enter the host cell. And this interaction determines also the species and organ specificity of the virus. Because if to enter the cell, the viral capsid protein need to match with the receptor on the surface of the cell. If it doesn't match, so it won't enter. For example, mainly bird flu virus cannot match with the human receptor, so that's why they won't cause infection. But now we know that some of the bird flu viruses manage to adjust to human receptor and enter. But then that determines the organ specificity. But also the antibodies against capsid proteins, for example, in rhinoviruses that are naked viruses. So the antibodies against these capsid proteins will protect us against infection, because as soon as they cover the places that's supposed to be used to bind with the human cell receptor, so they will not be able to do this. So these are the three very important functions of the capsid proteins of the naked viruses. But then if you take a look at the enveloped viruses, you can see that the capsid proteins are not expressed on the surface of the virion. So they cannot work as the places or as their receptors to bind with human cell receptors. So that's why in enveloped viruses, the capsid proteins don't have this function, the receptor function. And the receptor function is attributed to the envelope proteins, glycoproteins, because they are expressed on the surface, so they mediate the very first step of virus host cell interaction. And again, the envelope protein determines the species and organ specificity, and also those viral proteins, glycoproteins in this case, are responsible for entry, and that's why the antibodies need to neutralize or block not the internal capsid proteins to stop infection, but the external outer proteins, the supercapsid or envelope proteins. Then, how else we may classify the viruses? Of course, the very important classification is based on the structure and design of the viral genome. Again, it can be DNA, so we call them DNA viruses, so RNA, we call them RNA viruses, and the genome will be packaged inside a protein capsid in the virion. So, but these both nucleic acids can be designed in different ways. They can be single-stranded or double-stranded, made up of fragments or pieces, we call it segmented, or may appear as a single piece. And regarding the RNA viruses, they also can have the RNA of different so-called polarity. What do we mean under this polarity? So positive polarity is defined as an RNA with the same base sequence as the mRNA. And it means that the viral genomic RNA has 5' and cap. That is a starting point of translation and transcription. Translation. And fragment that is called poly A tail, right? That is working in a stop codon in the translation. And what does it mean? That positive polarity virus, so once their genome enters the host cell, it may be transported, the RNA can be transported directly to the ribosomes, and the viral proteins will be produced instead of cellular proteins on the ribosome. So from very first step of replication, the virus takes over the biosynthetic machinery, host cell machinery, for its own protein synthesis. But then, if you are talking about negative polarity RNA, it means it has base sequence that is compatible to, complementary to mRNA, but not identical. These viruses cannot give directly their RNA to ribosomes for translation. They need to convert first the negative sense RNA, the genomic RNA, into positive polarity RNA. My dear, that is of utmost importance. So, but then we discuss it a bit later on. So what else I would like to tell about the viral genome? So the viral genome usually codes for so-called structural, you see here, and non-structural proteins. For example, the genome of poliovirus is given, and these are structural proteins from VP4 to VP1, and these are non-structural proteins. So, sorry, genes that code for structural proteins and non-structural proteins. What do we call the structural protein that are translated from structural genes? So, these are those proteins that make the virion, so that are seen in extracellular virus particles, so that participate in building up the typical structure of infectious virus particles. But then we also may have a place in the genome for so-called non-structural or accessory proteins. What will be the function of those proteins? Of course, they are also very important in replication because mainly some information about some enzymes and quite helpful proteins is kept in these non-structural genes. But then, after these proteins are produced inside the host cell during the virus replication, and these proteins will full its function, then they will be not taken up inside the virion. They will be left inside the infected cell. So they will not be collected together and assembled in the virion. So that's why, and as soon as the virion release from the host cell, we won't see those proteins in this mature structure. So that are non-structural proteins or virus-encoded proteins. So then, you can see here the examples of DNA viruses and how DNA can be arranged there. And altogether we have around 6 families that are called DNA viruses. And here you can see RNA viruses with the different design of the RNA. It can be single-stranded or double-stranded, positive or negative polarity, segmented or non-segmented. But then it appears that RNA is more fashioned to be a viral genome than DNA, because altogether we have around 13 different families of RNA viruses. So viruses like RNA more than DNA. So, but then I would like to give you one more way of classification. It is not very typical in use in our country, but in the Western world, this is considered one of the most important way of virus classification. What is the basis for classification? Not just whether they have DNA or RNA, but also the design of DNA and RNA. Why? Because the design of RNA and DNA determines the strategy for genome replication that we have touched already a little bit. For example, so we have two groups, group 1 and 2, according to Baltimore virus classification of DNA viruses. And one group is for double-stranded DNA viruses that have just DNA of both polarity, positive and negative. And the example is, for example, Hepatitis virus, right? So then, how the genome of this virus is expressed. I hope you know what does it mean expressed. If the gene is expressed, then what happens? The DNA should be transcribed into mRNA, here you can see, and then mRNA will be sent to the ribosomes for translation. This is the most, the way closest for human way of gene expression, right? When from double-stranded DNA, the mRNA is produced and then the protein. But then we may have single-stranded DNA virus, right? And they enter the group 2. So, because the mRNA cannot be produced, transcribed directly from single-stranded DNA positive sense, right? So, because it may give rise to minus sense RNA only. So, what happens? What is the strategy for this virus for genome replication? They first build up the negative sense RNA, right? Negative sense DNA to make double-stranded DNA. That's it. And then from minus strand, the mRNA will be produced and then it will be sent for the ribosomes to transcription. Okay? Then we may have the way that is the typical for RNA virus or ways typical for RNA viruses. And here we can see more variability of the strategies. And the strategy depends on whether it is double-stranded RNA, like for example in rotaviruses, or single-stranded RNA, positive polarity like in polio, or single-stranded RNA like in negative. So that if we have double-stranded RNA, then there is no so much difference. So the mRNA will be produced from negative sense and then it will be sent for translation. Then if we have the single-stranded RNA molecule, this molecule is the mRNA. So it may be sent directly as mRNA for translation of the protein. But then to build up new RNA strands for the offspring, this positive sense RNA need to be converted to minus sense RNA, right? And then the genomic RNA will be produced out of it. Then one more group is arranged for single-stranded RNA viruses with negative polarity RNA. What is the problem? So their RNA cannot be recognized as mRNA. So first, positive sense RNA should be produced, and then this positive sense RNA will be sent for ribosomes for translation. And then we have two more groups over the viruses that may have reverse transcribing activity. So, and it means that they may build DNA using RNA as a template. That is unusual activity that is never seen in the human organism. So it needs the unusual enzymes, and this enzyme is called RNA-dependent DNA polymerase or reverse transcriptase, and the group is called retroviruses. When they need to express their genome, they first build the DNA based in RNA as the template, and then full copy of double-stranded DNA. And from this double-stranded DNA, the mRNA is produced that is sent for the ribosomes for translation. That is totally different strategy from what we have discussed before. And one more virus that may have reverse transcriptase activity, and it was placed in the last group number 7, and this is Hepatitis B virus. It is DNA virus, but its polymerase, DNA polymerase, has reverse transcriptase activity. So these DNA viruses, interestingly, replicate through the RNA intermediate. So when the RNA intermediate is produced, then the minus sense, then the positive sense RNA is produced and as mRNA sent to the transcription. Okay. So, and here you can see the examples of the different types of the viruses from different groups of the RNA or DNA viruses, and the full spectrum of the viruses families that may belong to the particular types according to the Belkin. Now we discuss viral replication. So, and of course, this is really quite difficult to understand from the first try. So that's why we just roughly discuss it, and then you read more accurately in the books, because you need to think and repeat several times to understand how it works. I just give some discuss some milestones of this way of replication. Again, that is the unique way of replication. And as I said, so the viruses usually cause the alteration of cell morphology when they multiply inside the cell. And usually it is accompanied with the disarrangement of the cell function and structure. So the process culminates in the lysis and death of the cell. So then, as I said, these viruses are called cytolytic viruses. But then the changes inside the cell can be not so dramatic. So, but irrespectively on the scale of the disarrangement of the cell or scale of the harm to the cell, the viruses that cause those changes are called cytopathogenic viruses, and the effect that they cause in the cell is called cytopathic effect or cytopathogenic effect. So this is any effect that is seen really. We may see it, what happens with the human cell under the light microscope when the process of virus multiplication occurs inside the cell. I think later on we touch it again, but then just by now that's it. So now, the process of viral replication inside the host cell can be generally categorized into the 6 stages. So, what happens during the first stage? The virus recognizes the cell receptor on the surface of the host cell and adsorbs to the receptor. First, this is the, usually the adsorption is highly specific. So then it follows with the stage of penetration. The virus that is outside somehow is transmitted to inside. So this is called penetration or entry. So the third stage is uncoating. What is it, uncoating? This is removing the proteins, or we may also call it deproteinization. And as a result, the viral genome starts to be accessible for the processes of transcription and translation. Sometimes we may have not the complete deproteinization of all capsomers from the capsid. No, some viruses hide its nucleic acid because it can be easily damaged by some human interleukins or cytokines. So then they keep it as a secret inside the protein shell. But anyway, it should be accessible for the transcription and translation process. The particular mechanism of it will be studied when we study individual viruses in special virology and the strategy of replication. But generally, you have to understand by now the most common strategies of replication. Okay. As soon as the uncoated happens, so the followed stage, but it is number 4 already, that is biosynthesis of macromolecules. What kind of macromolecules supposed to be produced? You can see it here. The protein should be produced on the ribosomes, right? And the nucleic acids, the genomes for newly synthesized viruses also supposed to be produced. At least 2 types of macromolecules supposed to be multiplied inside the host cell. So when the proteins are produced and nucleic acids are produced, so then it enters the stage of maturation and assembly. One word is missing here: maturation and assembly. So you can see the final result of it. The capsid is built and the nucleic acid is transported and enclosed into the capsid, right? So then the virion is done. Now the next stage follows, that is the release of the virus offspring, of the virions. And you can see here that one of the ways how it can be released, that is shown here, that is the rupture of the cell membrane. So this is the example of cytolytic virus that causes lysis of the cell. So of course we may have a lot of characteristic features of each stage. So I will mention some, but then I would like to draw your attention that this process is very complex and individual for distinct types of the viruses. So we just discussed some examples of it, okay? So when we have the, see, so that is the first stage. You've seen this picture already. The cell receptor should match with the virion capsid protein if it is naked virus, or glycoprotein C if it is an enveloped virus. See, if this is the enveloped virus, this is a nucleocapsid with the genome. By the way, the HIV is shown here. Then we have a sort of matrix protein, and then we have a lipid bilayer cell, like in the cell, right, but modified with viral proteins that are arranged in spikes. So, and in the spike we have a subunit, and the whole thing is called polypop structure, right? So, and the candy, this is glycoprotein 120. 120 is the molecular weight of this protein, of this portion. So, it coincides with the cell surface receptor, that is CD4 on the surface of the T helper, very important cells of immune system, right? Clue cells in the development of the immune response. So this virus has tropism for this receptor. So, and the interaction is highly specific. Okay, then the second way is penetration. Viruses generally use 2 basic ways. First way is so-called receptor-mediated endocytosis, when the whole virions bound with the receptor will be engulfed into the endocytic vesicle, and appear inside the cell in this vesicle. So this is receptor-mediated endocytosis. This is a picture, this is an electron microphotograph how viruses can be catched inside the endosome. So then another way is called fusion, that is typical for enveloped viruses with some exceptions. So because the viral envelope and the host cell membrane are very much same, so the virus, when it binds to the cell surface receptor, it may enter through the fusion of the viral envelope with the cell membrane. Usually this entry is mediated by some fusion peptides or proteins located on the glycoproteins of the envelope. So this way is called fusion, this way of entry. It is believed also that very small viruses may enter directly through the pores in the cell membrane, and this is the smallest papilloma viruses, but that is a question mark by now. Then the third stage is uncoating, and for example, for influenza virus, as we see here, it enters and there's an exception. This is the enveloped virus, but it enters through receptor-mediated endocytosis. See, this is the endocytic vesicle, and the whole virus appears here. But due to the acidification of the medium inside the endosome, the viral envelope fuses with the endosomal membrane, and the segmented influenza virus RNA genome is released from the endosome into the cytosol. So finally, the viral genome appears in the cytosol. That is the task for the virus, for the viral uncoating. Then the stage of biosynthesis occurs, and it is really very sophisticated in some viruses, and that is very good example in demonstration of the conjunctive way of replication, when the different fragments of the virus will be synthesized and assembled in different compartments. For example, in influenza virus, nucleic acid and capsid proteins will be assembled in the nucleus of the human cell. And then from the nucleus, this nucleocapsid will be transported towards the cell membrane. And the proteins will be produced in a cytosol, and some proteins will undergo glycosylation, for example, like hemagglutination and neuraminidase on Golgi apparatus. And only then, after biosynthesis, and the nucleic acids will be produced in the nucleus also. And then out of these, several types of components, proteins, nucleic acids and glycoproteins, we have maturation and assembly of new virions. And also, we'll discuss the more sophisticated way of maturation. You see that the viral nucleocapsids are assembled in the cell nucleus. Then they start to be transported towards the cellular membrane. But then the surface glycoproteins will be sent first from Golgi apparatus and modify the cellular membrane. And then M protein should line these small, things here. The M protein should modify this cell membrane from inside. What for? To make nucleocapsids to bind to be transported towards the not just cell membrane but modified cell membrane. Do you remember we have discussed the function of M protein? This is it. So the M protein demonstrates or shows to the nucleocapsids where to bind with, right? So it mediates the interaction between surface glycoprotein and nucleocapsid protein. So then the nucleocapsid will be taken towards the modified cell membrane, then evagination occurs, and then the newly synthesized virion will be cut off the human cell membrane. This way of release is called budding because it resembles the bud of the plants in spring. Spring is coming already. So then we have discussed two ways of release. This is the lysis of the cell, and this way of release that is typical for influenza virus and HIV virus is called budding. See, the buds are formed where in this false colored scanning electron microphotograph, the red things are surface glycoproteins, and you can see how they are had been transported, and you see that they modify the cell membrane. Then the membrane is lined from inside from white layer, that is M protein, and then the yellow things are nucleocapsid that are transported here and then cut off completely, you see here, from the cell membrane. So these are parts, so these are newly synthesized virions. And they may not kill the cell. See, the cells still feel alive. But then it will die anyway. And this is the most important property of the virus and way how they cause pathology in the human body. There are some other ways, and we will study them when we study separate viruses, the HIV virus, coronavirus, and others. But generally, that is the major damage that viruses do to the human. So you can see a more virus replication where there is the enveloped virus. It enters through the fusion with the cell membrane. Then mainly the viral genome transcription occurs in the nucleus because it is a DNA virus, it is very convenient. Then the proteins produced in the cytoplasm and transported back to the nucleus to make the nucleocapsids. And then the virus will be released by fetching a piece of the modified cell membrane by budding. So this is a typical for Herpes virus. Now a few words about the isolation, culture, and detection of animal viruses in the laboratory. So you studied already the culture method applied for bacterial infectious diseases diagnosis. When we recover bacteria from the specimen taken from the patient and with different tests, identify these bacteria. So generally, the sense of virus replication is very much same. We take a specimen from the patient, then we need to multiply the virus, but then usually, because it is difficult to see first whether virus is replicated there or not. So first we need to detect whether the viruses are growing in our model or not. And then if yes, if the viruses are there, then we have a stage of identification. So culture, detection or indication of the presence of the virus, and the third is identification. So this time we won't discuss identification, because for this you need to learn a bit more of immunology next time and special virology. But then when we discuss the particular viruses in the special virology, then we discuss the identification techniques also. So then, the problem is, how do we culture viruses? So we cannot grow them on the bacteriological media, so altogether three approaches exist how we may multiply the viruses. The most historically, the oldest one is animal inoculation. Even Louis Pasteur inoculated the rabbits with the viruses from infected rabbit dogs, right? So he didn't know that this is virus yet, but he had this organism in the brains of the infected dogs, and he passaged this virus through the organisms of the rabbits. So that was animal inoculation technique, right? When we inject or introduce the virus to the living body, and they develop in a body of the laboratory animal. Then the second way is to infect embryonated eggs. And the third way that is most applicable right now is the cell cultures. So step by step we discuss this. So that's very simple to discuss animal inoculation. We inoculated, then the first stage, second stage, detection. How can we detect whether the virus is growing in animals or not? So the first you see, the animal may die. Then that is a way of detection. Then we take the virus from the animal body and identify. But then sometimes animals may not die. They may develop symptoms of some diseases. Some general symptoms like dizziness, motionless, loss of appetite. That's also when wasting syndrome, when they losing weight. That also the symptoms or the signs of the virus multiplication in the animal's body. So sometimes it may be more obvious symptoms like lesions on the skin, or if you infect mice with the mouse-adapted influenza virus, they may start sneezing and coughing. So that is the way of detection. But then more common is the cultivation of the animal viruses in embryonated hens' eggs. So here you can see the so-called open way, when we partially remove the shell of the egg and inoculate the egg with the inoculum taken usually from the patient. And there are several ways how can we cultivate the animal viruses in the embryonated egg. You can see the several ways here, and they are attributed to particular types of the viruses. For example, this is a very good way for influenza virus to grow, or for Hepatitis virus or pox virus. So, when the viruses are growing in the embryonated egg, so how can we detect the presence of the virus? So if we inoculate it directly on the chorioallantoic membrane, then the viruses may produce visible lesions that are called poxes. So under optimal conditions, each infectious virion causes the formation of the one pox. So we may use it as not only as quantal, but also quantitative way of the detection. But still, even So we can see these boxes. We detect that were, for example, 1, 2, 3, 4, 5, 6 viruses in the inoculum, but still we don't know exactly what species of the virus it is. So the next stage will be identification, right? So then, other than chorioallantoic membrane, so we may culture the animals in the allantoic cavity, in the amniotic sac, or in yolk sac. Then the problem is, if the virus doesn't cause any visible changes in the embryonated egg development, and that's quite common. So how, after 3 days of incubation, we can understand whether they are growing there or not? For this, we may use some simple reactions, for example, hemagglutination reaction. So the classical example of the virus that can be detected by this way is influenza virus that has the hemagglutinin spike on its surface. And this hemagglutinin spike may bind with the surface of erythrocytes. So when by this, the influenza virus may cause clumping of erythrocytes. That is called hemagglutination. So, but then when we culture the virus, for example, in allantoic cavity or amniotic sac, then after 3 days we suck out the allantoic fluid where we're supposed to have some viruses if they grow, and then we combine this allantoic fluid with red blood cells. And this allantoic fluid, if it contains viruses, then you can see here that viruses will cover erythrocytes, and through hemagglutinin, all erythrocytes will stuck together under the gravity when they fall down the bottom of the plastic well in a plastic plate. So then they drop not one by one, but all together to form like an umbrella or net. That is called positive hemagglutination reaction. And you can see here, if the erythrocytes are not aggregated with the help of virus, if there is no virus in the liquid, so then they fall down the bottom of the well under the gravity one by one to fall like a pile of the erythrocytes, so it will be seen as dot. So that is the difference of the positive reaction that we have here and negative reaction. And we also may perform titration of the virus containing liquid to see in what dilution we still see the positive reaction. So for example, this is the last dilution when we see positive. The next one is negative already. So this dilution will be called the titer of the reaction. So if these are two full dilutions, 1 by 2, 4, 8, 16, 32, 64, 128, 256. So the titer will be 1:256 dilution, right? So that will be last positive last dilution where we see positive reaction. That's how we identify the amount of the viruses in this virus containing liquid, okay? So that were about the detection of the viruses when we grow them in the chicken embryo. But then the most convenient and common way right now in cultivation of the animal viruses is the growing of the viruses in tissues or in cells. So initially, quite a lot of models had been done with tissue cultures. So for example, the organ culture will be done to identify to isolate coronaviruses, and the tracheal ring organ culture had been performed. And in this culture, the organ will maintain its normal structure for a while and function. Then they also explant cultures can be developed when a piece of the organ will be cultured in the surrounding protein or serum containing liquid. For example, that way had been used for isolation of adenoviruses from adenoid tissues. But then the most common right now is the true cell culture. So when we culture not the whole or piece of organ, but the particular cells. And usually the cell cultures can be divided into the three categories, according into the three types, according to the origin of the cells, number of generations through which they can be maintained, and the chromosomal characters. So when the first type is called of the cell cultures, it's called primary cell culture. Why it is called primary cell culture? Because these are cells freshly taken right now, taken from the body and then maintained outside the human body. So usually these cells are capable of limited growth and can be maintained for 7 days. Why? Because they will not divide outside the tissues, right? In vitro. This is in vitro growth. So they stay alive, but they cannot divide. So we may maintain it for about a week, but that's enough because usually if we infect the cells, the viruses will be produced, new viruses will be produced inside the cells within around 3 days. So here you can see the general way how we produce this culture. So for example, we take the organ, cut it, then finally cut tissues or explant, and then we treat it with trypsin to get it finally cut and to let the cells to come out of the stroma, right? So for particular limited time. So, and because we use trypsin, those primary cell cultures are called trypsinized cultures. So then we wash it out of trypsin and separate through the centrifugation. So we divide the single cells and the cell debris and the pieces of organ. And then we place these cells into the liquid that contains proteins, amino acids, buffer solution, and this liquid is usually called culture medium for eukaryotic cells. My dears, this liquid culture medium is not to grow viruses, but to grow eukaryotic cells that can be infected with the viruses, that will produce new virions, okay? So those cells may be placed in monolayer or in suspension. But then the idea is that those cells are used as the targets for the viruses to be infected and to produce new virions. So now the second type is diploid cell cultures. And these are the cells of single type that retain the original diploid chromosome number, so they can divide, but they may divide a limited number of times, right? So they may undergo about 50 passages. Then they undergo senescence and die, those cells. So we may maintain them within not more than 5-6 months. Then they die. But this is quite convenient because we may maintain these cells alive, placing in the culture medium by serial passages, not for just a week, but for half a year, for example. And here the example of this cell cultures. And that's the cell culture in common use. There's a diploid fibroblast. But then we have also the third type of cell that can be maintained. And first two types are normal cells. And this, the third type, these are called continuous cell cultures. Why? Because these cells never die. That's why this cell culture can be maintained by continuous serial cultivation for many years, for decades. Hera, that was the first cell line developed around 60 years ago, 6 decades ago, sorry. So, and then from human cervical carcinoma. So, and then still any virological laboratory in the world may keep those cells alive, right? So by serial subcultivation, if we don't need these cells, we may freeze them under -70 degrees Celsius, and then if we need them, we unfreeze them and place it into the culture medium. For eukaryotic cells, they start multiplication. Those cells are tumor cells that lost the contact inhibition, so they grow without any limits. And you can see here, when normal cells are growing, so they grow like this, they form monolayer. But when they transformed or tumor cells are growing, they will not growing as monolayer because they lost the contact inhibition. So, we have discussed several types of culture media or cell cultures that are used for the growth of the viruses. But then now the question is, when we infect the cells with the viruses, so we keep them there for 3 days. Then how can we know? How can we indicate whether the virus is growing or not? For example, for those viruses that can cause visible changes inside the cells while multiplying there, we may see so-called cytopathic effect. So because the viral growth produces a characteristic effects or morphological changes in the cells. So cytopathic effects can be different, but usually they can be absorbed under the light microscopy of the cells infected with the virus. For example, we may see cell rounding, sorry, no, not that, cell vacuolization, see, appearance of a lot of vacuoles, and this is small magnification, this is higher magnification. So vacuolization occurs when the components of the virus start to be transported within the cell. the cells may undergo cell rounding or losing their normal shape. Then some viruses may cause cell lysis. Then that also will be considered as CPE. And also, because of the formation of new proteins inside the cell and accumulation of these proteins and viral structures inside the cell, we may see it as the inclusion, unusual inclusion body formation inside the cells. This is low magnification and this is higher magnification. So after staining, we see the unusual inclusions of the rabies virus cells in the brain matter. So that can be used in the diagnosis of this. But then if we have the viruses that multiply inside the cytoplasm, then we may see the intracytoplasmic inclusion. But sometimes we can see, not shown here, the intranuclear inclusions. That's also possible, for example, for Hepatitis viruses. One more type of cytopathic effect is the sensitive formation, where the virus-infected cell may fuse together with non-infected cell to make giant cells made up of many cells and usually with many nuclei inside. And measles virus, HIV virus, respiratory sensitive virus may cause this type of cytopathic effect. But then the problem is that not all viruses may cause cytopathic effect. So some may multiply, like for example, rubella virus may multiply without giving for a long time any visible changes in the cell. So then how can we see whether the virus is multiplying there or not? We may perform the reaction that is called hemadsorption when we indicate the presence of intracellular virus, not extracellular, my dears. Hemagglutination is the indication of the extracellular virus, so over the virions, while the hemadsorption is the indication of the presence of the intracellular virus. When the virus multiplies inside the cell, at the particular stage of replication before the release of newly synthesized viruses, so the surface glycoproteins, the envelope glycoproteins will be expressed on the surface of the infected cell. Then if it has hemagglutination activity, these spikes of the virus, then if we add the erythrocytes to virus-infected cells, and there are hemagglutinin spikes on the surface of the infected cell already, so the erythrocytes attached to this cell, see, this cell is not infected, erythrocytes didn't surround this, didn't absorb at the surface of this membrane, cell membrane, but they absorb here because there are hemagglutinin proteins here. So this is a small magnification, this is a large magnification. So, and this reaction is called hemadsorption, where the erythrocytes adsorb to the surface of the infected cells covered with the viral hemagglutinin protein. So we also may use immunofluorescence to demonstrate the presence of the inclusion bodies. And that was for cytomegalovirus, and this is for Hepatitis virus. My dears, the next is bacteriophages. It is not so difficult. And you can see here the picture where bacteriophages binds with the bacterial cell, infects it, multiply there, new bacteriophages are produced and rupture the cell to release. So this way of replication is very typical for the animal viruses. And this is called bacteriophages Lydic cycle. When the bacteriophage injects its nucleic acid, multiply within the cell. So then maturation and assembly occurs, and the newly synthesized bacteriophages rupture the host cell. The cell undergoes lysis. But then we may also have bacteriophages with a different life cycle. This is called lysogenic cycle. So the initial stages of infection are safe. For example, the bacteriophage enters the host cell or injects its nucleic acid into the cell, but then the nucleic acid of bacteriophage, if it is double-stranded DNA, may integrate with the bacterial chromosome. So it is ready, this here. And because the ability to multiply is depressed with the repressor protein, then the virus doesn't multiply itself with the help of bacterial biosynthetic machinery. But then when the bacteria divide, and before dividing, binary fission undergoes the genome replication, of course, the bacteriophage genome also will be duplicated. So the daughter cells will get also the bacteriophage genome. So that's how the lysogenic bacteriophages may spread within the population of bacteria. That is called lysogenic cycle. But then we believe that from the lysogenic cycle, bacteriophages can be activated by UV light, for example, or some chemicals. Then the repressor protein that keep under the control the viral genes replication. Then it will be removed, and then this virus may enter the Lydic cycle of replication. So the proteins and viral genomes will be produced in a cell, then maturation and assembly occurs, and they release the new bacteriophages by cell lysis. Why this event is important in medical microbiology? Because those bacteriophages that undergo Lydic cycle may be used as the drug for treatment of some bacterial diseases. For example, we may have the preparation that is called enterophage that will cause the lysis of the enteric bacteria Escherichia, Shigella, Salmonella that cause the particular diseases. And from another side, and some more ways of application are possible. But then from another side, the genome of the bacteriophage may keep some genes that code for some proteins toxic for humans. We may call them exotoxins. If bacteria are so-called lysogenized or infected with lysogenic virus, it may start producing some new proteins. For example, the exotoxins that can cause the, for example, lysis of the human erythrocytes. For example, this is hemolytic properties can be hidden or encoded by the viral genome. or antibiotic resistance also can be transmitted with the help of the bacteriophage genomes. So that's why this event, that is called lysogenic conversion of the bacterial cells by bacteriophages, is very important also in medical microbiology. Guys, we managed to discuss all features, all that we planned for today. With this we finish. Goodbye, my dears. See you in the classes. The lecture is over. Goodbye.